Hello. In this biology lab, I want to go over the parts of a microscope and how to get an image in focus so that you can see what you need to see. This is a compound microscope. It has ocular lenses here that will magnify the image 10 times. A stage here where the slide goes. It has a clamp to hold the slide in place. There are knobs here. The first one moves the stage up and back and forth. The second one moves the clamp right and left so that you can center your image under your objective lenses. This microscope has 4 objective lenses. The first one has a red stripe on it. It has a magnification of 4. The second one has a yellow stripe on it, and it has a magnification of 10. The third one has a blue stripe on it. It has a magnification of 40. And the fourth one has a white stripe on it. It has a magnification of 100. There is a small lever under here called the iris diaphragm that controls the amount of light hitting your specimen. You can close it or open it depending on how much you need. The power switch is on the base here. This large knob is the coarse adjustment knob. It will raise the stage up quite a bit. The smaller knob inside it is the fine focus adjustment for getting it in focus for your vision. These microscopes are parfocal, so that means if you get one lens in focus, you can switch around to another lens. It should still be in focus. The easiest way to get these in focus is to put the yellow lens pointing down. I have a slide here labeled E, and we're going to try to get that in focus. I have it hooked up to a computer so that you can see what I'm looking at. And at the moment, it's not doing anything. That's interesting. Okay, we'll close that one. Try opening this thing up again. Now, I will use the coarse adjustment to raise this stage as high as it will go. Will not hit the slide with your objective lens, I promise. And there is my specimen. It's a little bit out of focus, and it's a little bit bigger than I want. So I adjust these knobs to get it in the center. Then the fine focus, I turn it until it is in sharp focus. That is in sharp focus for me. Now, since it is taking up a big portion of my screen here, I can shift it to a smaller lens, and you can see the entire object. This is an upside down backwards E. The reason I show this, it's easy to work with, to get comfortable with the microscope, but it also illustrates that the image, having gone through multiple lenses here, gets flipped multiple times, so that by the time you look at it, it's upside down and backwards. If your partner is standing out here to the side and looks at that slide, the E looks right side up. You looking through the microscope, it's upside down and backwards. So which one of you is correct? It turns out both of you are, depending on your point of view. So again, that is an easy thing to work with just to get comfortable with these microscopes. I'm going to try a second slide. And all I did was lower the stage, move the slide out, put a new slide in, raise the stage up as high as it will go. Let me put the yellow lens back down again. And I can see a little bit of green there. So I will move the slide again. That's what I'm looking for. I have 3 green threads here, and I'm going to have to use the fine focus to get them in focus. Now for me, that green thread is in focus. A little bit more adjustment and the red thread is in focus. Go all the way back to here to get the yellow one in focus. I cannot get all 3 of them in focus at the same time. This is because we have the depth of field. We have to remember that third dimension. When we're looking at specimens this way, we also have to remember to look at them this way. Those threads cannot get in focus at the same time because they're not on the same level. There's one on top of the other. And when you focus on the top one, the bottom one is not in focus. So you have to focus on the top one, then adjust that fine focus again, that looks like the green one is in focus for me. I turn the fine focus in one direction, I get the red one in focus, I continue to turn it till I get the yellow one in focus. So the green one is on top, the red one in the middle, and the yellow one on the bottom. And I have to adjust that fine focus all the way up and down to go deeper into my field to get that one in focus. So that is a quick demonstration of how these microscopes work, how all the parts work, and how you get some specimens in focus so that you can see what you need to see.